Since you've been so good and since it's your birthday, here's a little extra thing on some RNA virus polymerases that we might not cover otherwise, and it's extra fodder that you can throw at your RNA polymerase function answers. Yeah, so come on, let's get into it. Okay, so I'm going to cover three other groups of RNA polymerases, because why not? As a representative of the fourth group up above us, I wish it was an animated GIF. I really do, but it's a cool picture. I believe that one is the poliovirus polymerase up there. And that's fine. We'll cover that in class. Here are the extras. So the three we're going to cover are one from a flavivirus. So that's going to be like yellow fever or dengue or Zika or hepatitis C. One of them from a levivirus. Those are bacteria infecting RNA viruses, which totally exist, and there are tons of them, and people generally didn't look for them in the past and are only now starting to realize that, hey, yeah, there's a lot of weird stuff out there. A famous one would be called Q beta. That's like the king of these. Or queen. Yeah, I don't know. Yeah. Not have to get weird about it. Let's see. The third one to cover is a double-stranded RNA virus. And so this is going to be a reovirus, similar to something like blue tongue virus, but there's a bunch of other reoviruses. Okay, let's get into it. So here you have the beautiful flavivirus RNA polymerase. So the protein itself comes in two parts. They're called NS3 and NS5. There's an NS4 in between. We think it comes together and forms a kind of a scaffold to help hold these things together, but I don't believe anybody's solved the structure of all of those yet. And I think NS4 has some hydrophobic parts, if I remember right, but don't quote me on that. So anyway, NS3 is mostly going to be the helicase in this case, and it's going to do two jobs, just like the coronavirus helicase. It's going to have a triphosphatase, so it's going to take the last phosphate off of a nucleotide, just like terminal phosphatase would do. Yeah, hint, hint. And it's going to be a helicase, so it's going to unwind, probably peeling the template off from the thing that you just copied, so that you can then have another polymerase situated right behind it that then makes a copy, and so you don't have to wait to make like a complete copy. You can actually have maybe 100 polymerases running off copies at the same time in glorious, delightful chaos. And so that's the NS3. NS, by the way, stands for non-structural. Like in coronaviruses, we call it NSP, non-structural protein. In flaviviruses, they just call it NS. Whatever, deal with that. Yeah. The polymerase is in NS5, and NS5 has two big parts. NS5A, which is a capping enzyme, and NS5B, which is a polymerase. So in the image in front of you, the polymerase is kind of doing this, or rather this, I guess, if you're looking at it. Yeah, there you go. I was showing it to myself, not the most effective way to show something to someone else. And so I think the fingers are on the top, but I would have to look at the structure in some other form to actually tell the difference. But yeah, the palm is definitely on the bottom. The active site is this little blue thing, and that's where we're putting in new nucleotides. So congratulations. The other part, though, of NS5 is this capping enzyme. And by capping enzyme, I mean it does 3 out of the 4 capping steps. Remember those steps? Terminal phosphatase, the helicase does that. Then you have guanylyl transferase, which is putting the backwards G on. And so this thing will actually do it the way a cell does it. It's going to have terminal phosphatase chop off one phosphate here. It's going to get a GTP rather than a GDP, and it's going to join that on and release 2 phosphates. 2 extra phosphates are going to be kicked off of there. And so it can put the cap on, neat, but what else can you do, NS5 capping enzyme? Well, it turns out this is a methyltransferase. And so it has S-adenosyl methionine, that's our SAM here, and you can see it written out down there, S-adenosyl methionine, yeah, which is the methyl donor on methyltransferases that modify RNA. I think exclusively, as far as I know. There may be one out there that works a different way, but this is pretty much it. So we're going to get methyl group off, and we're going to stick it onto the cap. Awesome. But we think this one actually does both steps. So it will both methylate the first nucleotide, that N7, and it will then pull it along and methylate the second nucleotide, which is the 2' O methylation. Yeah, seems to work like that. So, fantastic. It's kind of a one-stop shop in that it can put the cap on and methylate up the cap, and bam, you've just made yourself a cap. Once again, why didn't coronavirus think of this? It's not so hard. Yeah, corona's carrying around 4 separate enzymes to do the 4 parts. Yeah, ridiculous. Yeah, ridiculous way to run a virus. But here you go. Okay, so that's flavivirus. That's the process. That's the machinery. Kind of similar, right? Yeah, you're seeing the general trends, and I think these general trends really only become apparent when you're looking at a couple of these viruses. So, can you spot which one of these things is the polymerase? Let me give you a clue. The polymerase will have a palm that has a beta-stranded floor on it. Like, ooh, maybe. Even if it didn't have that word there, you could see, oh, look at that. Bunch of beta strands lined up in a little curved beta sheet. Looks like a palm to me. Yeah. It's going to have a sort of a thick side with lots of alpha helices sticking up, and so those are going to be the fingers. And it's going to have a sort of a thinner side to that, and again with alpha helices, we call that the thumb. It's not really that different from the fingers in appearance, I will agree. And then this one has an extra bridge which sort of closes it off, which is a weird thing to do. Usually there's another protein that does that job. So there you go. That is the polymerase of the Q-beta bacteriophage. Now, bacteria are different in that they don't have to cap RNA because bacteria translate RNA that has no cap whatsoever, no cap requirement. The idea is that the requirement for a cap is probably a hedge against filthy viruses that are coming in and, like this one, just running off uncapped transcripts and hoping they would get translated. Eukaryotes leveled up and never looked back. Since then, we've just been adding methyl groups on and taking names, basically. Yeah. Eukaryotes, Team Eukaryote for life. There you go. In this case, though, what it's going to do is it actually interacts directly with the EF-TU complex. So this is a little thing that's going to be clipped onto the ribosome. This is an elongation factor. So this is something that the ribosome is going to need in order to continue working. And the virus is very deliberately clipping its polymerase on. The polymerase actually, the active site collapses if it's not, or it's not in the right position to work, unless it is attached to these. I believe that is the full story. And so by attaching to this component, which is essential for translation, you do 2 things. One, you open up the polymerase for business, so you turn it on, turn the machine on, just like hitting the button here on my laptop. And the other thing is that you position the RNA as it comes out. It's already on a component of the ribosome, so you better believe that thing can get translated quickly. And Q-beta is a fast little virus because it cuts out the middleman completely. Yeah, it's just, here's my RNA, and while I'm making the RNA, why don't you get to work translating the RNA? It's that efficient, that level of efficiency. So really cool little virus. But you can see the polymerase part, kind of the same. So just a different way that we're actually going to use this same machinery. And that is the theme all the way through this. So this is a polymerase over here. Just looking at it, can you spot where the different parts are? Can you see a big beta strand that would be the palm? My palm has something written on it. There, the palm. And yeah, look at that, right down there on the floor. Big little curved beta strand, like that, to hold the RNA. The blue and orange are the RNA template, I believe, is blue, and I think the new RNA that's being made is the sort of rusty orange color. And then the green is the protein, and so you can see a ton of alpha helices over on this side, and so that probably is the fingers. Yeah, if you're scoring along at home, and you can see a side that has fewer alpha helices, and that is probably going to be the thumb, although it's possible that I have those two reversed, but yeah. they're both there, and they're the two sides. They're the two things either side of the polymerase, because the polymerase is always doing this. It's always making this gesture, basically. Yeah. To make a point. Yeah. To make an RNA. Yeah. In this case, the reovirus actually is multi-segmented, so sort of like influenza virus. And so each of those segments is going to be packaged with its own polymerase. And so these little pink patches are the polymerases inside the virion. They're loaded at specific strategic points inside that virion. And they managed to get a real nice structure of them. And you can see this sort of twisty stuff, that's the viral genome. That's an RNA double helix, double-stranded RNA helix. Pretty cool resolution, yeah, from electron microscope pictures. And then here you can see one of those sort of twisty yellow strands going in and actually pointing into the polymerase. So this virus comes ready for business. Each one of its strands has a polymerase on it. With the strand positioned in the polymerase, all this thing has to do is enter a cell, find a way to get some, not nutrients, but nucleotides and a little ATP to make the machine go, and then it's off. It is ready to start copying itself. And yeah, blue tongue is a pretty bad disease in that it's really easy to spread and pretty hard to shut down. And it's kind of a problem. It's one of these things that is, at least at current telling, a big enough problem that I think the U.S. doesn't even try to control it. In most other parts of the world, they do. And so that limits, you basically wouldn't be able to send certain kinds of animals from the U.S. outward, if I have that part correct, because they may have blue tongue, and there's nothing you can do about it. But the U.S. could potentially import animals from everywhere else, because most other places do make an attempt to control blue tongue. So, yeah, neat. Yeah, and so that's how it works. But you can see, once again, these are roughly the same polymerase, and they have different strategies for how they work. They have different little accompaniments. Some of them hold on to a bit of the ribosome, like our little Levi virus here. This is Q beta. Some of them, like the Zika virus polymerase over here, are going to have a helicase that's attached and a methyltransferase, guanylyl transferase little factory that will do a whole bunch of the steps of capping. And so they can just cap it as they make it. Yeah, easy peasy. Real nice. All right, there you go. That's a little extra about some RNA polymerases so that you can start to see the trends here. So go back through, maybe look for some notes, maybe pull out some details, formulate your answer in the form of a very nice essay answer, and you're going to be set for the next test. All right, good luck to you.